Hi, this is Mr. McLean, and in this session, I'm going to take you through the early parts of Chapter 9, which gets us into cellular respiration. I want to spend a lot of time making sure that everybody's good with the idea of the redox reaction and how that relates to cell respiration. We'll also be addressing either in this section or in the next section the process of glycolysis followed by conversion of pyruvate, the Krebs cycle, and the electron transport chain with oxidative phosphorylation and chemiosmotic ATP production. So we have a lot of things that we want to look at for Chapter 9, and we want to first kind of jump to, let's start by looking at what's happening with cellular respiration. So if we look at it, we're talking about something where we're going to have an investment of a glucose molecule, and we're going to have oxygen being used as an oxidizer, and as byproducts we're going to produce carbon dioxide, water, and the main idea of this process is for us to generate energy primarily in the form of ATP. We also will produce some secondary sources of energy such as heat as part of that process there. So if we look at the idea of redox, the word redox indicates that there's two different paired reactions. We have an oxidation and a reduction. So for any of these types of reactions, you're going to need to have both of them present. So the acronym that I often like to look at is the idea of oil rig. Okay, so if you think about it, oil stands for oxidation is losing. Reduction is gaining. So whenever you're looking, and what they're talking about losing or gaining would be the electrons. So whenever we have a molecule that has been oxidized, it has lost its electrons. When we have an accompanying molecule that has been reduced, it has gained the electrons that came from the oxidized molecule. So throughout the Krebs cycle, in particular, we see a lot of redox reactions. We also see that during glycolysis. So cellular respiration utilizes redox reactions to kind of transfer those high energy electrons from organic molecules and eventually utilize that to produce ATP. That's our main objective here. So just to reemphasize what I just talked about, oxidation, a substance is losing electrons. So if something is oxidized. Now, one of the things that becomes a little bit confusing is the way the wording is taking place. So let's hit the second one here. If something is reduced, that substance is now gaining electrons. Easy way to think about this. If we think of reduced, we're talking about we're reducing the amount of positive charges. So therefore, we are ending up with something that's going to be more negative. So the reduced molecule has actually acquired additional electrons. It has reduced its charge. It's become more negative. Just a way to think about it. So what I was speaking of as far as the confusion with these things is that the substance that oxidizes something is an oxidizer. An oxidizer ends up reduced. So for example, probably one of the most important oxidizers that we could potentially talk about would be oxygen, a molecule of oxygen, O2. It's a very, very electronegative molecule, and it's a strong oxidizer. So it's going to take electrons from other substances. And when it does that, it's going to acquire electrons, and it's also going to acquire hydrogen ions or protons to go along with that. So we would say that oxygen is an oxidizer and it becomes reduced. So you just kind of have to think about what is being the oxidizer and what's being the reducing agent, and ultimately what each one of them are going to become. All right, so your book looks at a couple different little scenarios here. It says if we think about something like an ionic formation, we have sodium and we have chlorine. And we all know that sodium will form a positive ion. So sodium is going to give up electrons, and therefore we would say that that has been oxidized. So we get our sodium, and that's why it has a positive charge, because it's lost electrons, and the chlorine has gained the electron. So the chlorine has been reduced. Another way of looking at it is if we use these hypothetical substances, substance X and substance Y, and if we think about it, if substance X is being oxidized by substance Y, the electron that it has is being transferred. So that's why when we look at it after the reaction, we can see that substance X is missing one of its electrons, and substance Y has acquired it. So we would say that X is oxidized and Y is reduced. Now, another way of speaking about that, we could say that Y is the oxidizer. The oxidizer is the substance that steals the electrons and therefore becomes reduced. And we could also say that X is my reducing agent because it gives up electrons so that Y can become reduced and X is going to become oxidized. So it's a little bit confusing with that, but like we said, the electron donor is the reducing agent. The electron receptor is the oxidizing reagent. Now, we can look at some other examples that kind of go along with this concept. Let's look at the main idea here, which is the cellular respiration formula. So we start off, and we see this over and over and over again. We have our organic molecule. And this organic molecule is obviously going to be glucose. So in the overall respiration process, glucose is ultimately going to become oxidized. What is the oxidizer of the system? Well, there's a lot of things, intermediates, that are going to be intermediate oxidizers, but the ultimate oxidizer, when we look at the overall reaction, is oxygen. So oxygen is going to acquire the electrons, the high energy electrons that came from the glucose, and in the process of not only getting electrons, but it also gets hydrogen that goes along with that. So we would say that the oxygen has become reduced. And in the process of becoming reduced, it has become a molecule of water. And when we go through here, once that glucose molecule has been fully oxidized, it is now going to become carbon dioxide. And when we look at the respiration process, we're going to see at multiple points that after carboxyl groups have been fully oxidized, that we get this CO2 coming off kind of as a waste product. So it's important for us to be able to identify that and to be able to see those things. Okay. And, you know, if we look at this scenario right here, this molecule is very important in our respiration process. That is NAD, and NAD is an oxidizer. Okay, it's a strong oxidizer. And what it's going to do is, all right, here's the component of an organic molecule. The oxidizer is going to take electrons from that molecule. And what that's going to do is it's going to leave us with a substance that has been oxidized. And it's going to give us a molecule that is reduced. Okay? So you think about this. Why is this reduced? Well, it has electrons. It has acquired electrons. And in the process of getting those electrons, it has also acquired some hydrogen. Now, as we all know, and we see as we move through the chapter, NADH plus H is an energy intermediate. Okay? It's not directly used to drive cellular processes like ATP, but we will use NADH plus H to deliver high energy electrons and hydrogens so that when we do full-fledged oxidative phosphorylation, we should be able to make maximum ATP. That's our objective and that's our goal. Okay? So if we think about it, this particular slide is telling us that when we have cell respiration, glucose and other organic molecules are broken down in a series of steps. So the reason why we have so many different processes that make up the respiration, the overall respiration process, is that it happens in intermediate steps. We don't have any massive breakdowns and releases of energy, but we have it kind of taking place in a series of very enzymatically controlled reactions. And what we find is that the electrons from the organic compounds are usually going to be transferred to this oxidizer, NAD. Now, we also have some other oxidizers that show up. So, for example, when we get to the Krebs cycle, we see FAD, which is a close relative to the NAD molecule, also serving as an oxidizing agent. So something that would be able to grab and take hold of those electrons. So it says, as an electron acceptor, NAD is an oxidizing agent during cellular respiration. So another way of thinking about it is that once NAD has acquired the electrons and the hydrogens, it now is reduced. Okay, and a lot of times we see NADH plus H plus because it shows you that it's actually acquiring two of those hydrogens. But this represents our stored energy. So this reduced molecule is yet to be fully tapped, but when we do tap it, we'll be able to produce, hopefully, maximum ATP. Okay, so this picture right here just kind of gives us an image, a very, very small image, but it shows us how the nicotinamide in the oxidized form ultimately can become the reduced form. So this is the NAD plus version, and we're going to see that in the process of it becoming reduced, it's going to acquire hydrogens and it's going to acquire the electrons, and we see this dehydrogenase being listed as an enzyme that will take place in and help drive this redox reaction. So that's going to catalyze that redox reaction. So now when we look at it, we have NADH plus the extra hydrogen that comes through here. So this is the reduced form, and this is the form that has energy potential to do something for us. So we'll continue on with that as we move through. So the idea is that NADH is going to pass electrons down the ETC, or Electron Transport Chain, and it's going to do it in a fashion that's going to release the energy in a controlled mechanism. Okay, so instead of a single very, very exergonic reaction that could potentially damage the cells and potentially be explosive, what we do is we go down and we yield the energy in small packets so that it's more it's more easily controlled and it can drive reactions. I like to think of the electron transport chain, I'll go over this a little bit more later, as almost like a stairwell, in the fact that the electron is going to move down the stairs. And if it was a real set of stairs, what would move a ball or something else down the stairs would be gravity. Okay, so you have this gravity at the bottom. In the electron transport chain, it's not gravity that's pulling it down. Actually, back to here, it's not gravity. What we want to have is we have to have a strong oxidizer. So when we say oxygen pulls electrons down the chain in kind of this energy yielding tumble, sort of like what we see right here, this electron transport chain is being driven by ultimately redox reactions. So that's why we talk about it being so critical. And when that oxygen is eventually picking up the electrons and the hydrogens, like we see right here, we end up with something that is now reduced.